Okay. I think we're live, boys and girls. Who do we have?
Looks like we have Scott Duke and you in the live chat.
Yeah, everybody knows you guys. So, yeah, there's 2 watching, 4 waiting, apparently. Anyway, of course, some of those are probably us. There we go. Cool. I didn't have the sound turned on this time. Okay, so we're going to go over a little NephilimFree video.
Mm-hmm. Yeah.
Where Andrew tries to get NephilimFree to define information once and for all. And I'll share this. Hang on a second. I'm going to put my hand.
Trying to get him to answer the bristle question.
The bristle question.
Yep. Which, if people have watched some of our other streams, they might know what it's about.
It's possible. Nobody pays attention to us.
Yeah, unfortunately.
They hate us. Okay, let me see if I can hear you guys through my headphones. Let me just say something. Nope, can't hear you at all. What the fuck?
Well, nobody was talking.
Oh,
can you hear now?
An alternative to StreamYard that actually works.
Wait, what? Can you hear us or no?
Okay, you guys talking?
I'm trying to. If I had to guess, he hasn't gone into the cam mic settings and switched his audio output. Hello, Speed. Welcome. Dude, boomer.
Fuck. I think you went to another dimension for a second.
No, my Logitech headphones decided to connect with some device probably out there on the freeway somewhere instead of connecting to my fucking computer.
Is it working now?
Yeah, I had to shut them off and turn them on again and wait for it to reconnect. And then the test music on StreamYard, it's a little Christmas song. It was kind of cool. So I decided I think I'll keep StreamYard for another month because they played that cute little Christmas song. I was so impressed. Do you think? Okay. Share screen. And hopefully, let's see. Prove it to me. Prove it to me. Oh, my fucking cocksucker. Unless you've gone to the tab like currently, and unless you have another tab open on Chrome and you go back to it, Chrome can't find the goddamn tab, or StreamYard can't find the tab. It is a real pain in the ass. Yeah. They don't really give a shit about the people that are paying them like $30-$40 a month, and I'm getting a little tired of them.
Yeah, you should just get a OBS thing set up, and then you can do it through like Zoom or Google Hangouts or something.
And save $40 a month on this piece of crap. It'd be kind of cool. Anyway, so I'm not sure if I'm at where I'm at. Hang on a second.
It's muted.
Yeah, it's muted.
I hear the whistle.
You heard a whistle?
Yeah.
That's a good sign. That means we're live with Nephilim.
The Nephi Express is rolling into town.
Express.
Hey, Leo. Thanks for joining us.
Mm-hmm.
Okay. Where do you come in?
Somewhere, let's see. I think it was like 3 hours, 51 minutes or so.
Okay. Yeah.
Wait, so does he just chill in there like by himself for 2 hours before anybody else comes in and talk to him?
Yeah, he was for an hour after you went away. He was playing a game without saying a word. An hour.
Either that, or he'll just monologue, and by monologue I mean repeat the same line over every 5 minutes for an hour and a half.
Dude, Nephi has like 8,000 subscribers. I just noticed that.
Well, you also have to remember he's been around since like 2009, 2008, 2009. He's been around for a while. That's why Praise has 12,000 subscribers, yet maybe at max 15 people watching his live streams. It's the same with Nephi.
Wait, Praise has been around for a long time?
Praise has been around for roughly the same amount of time Nephilim has.
Really? I had never heard his name before like the past 6 months.
So Scott Duke's dogs are going to go crazy because of Nephi's whistle.
Oh, yeah.
Hate to see it.
There's something we can't say on this stream, and it has to do with a dessert. It's kind of like a Jell-O-like dessert, and We can't say that name 3 times. We have to be very, very careful.
Yeah, it has a beigeish tan color.
Yeah. Okay.
The Yuki also. If you want.
I seem to remember the name Bionic Dance. I don't remember the other. I don't.
She's a QL. Yeah. Someone died.
So, oh, it was okay. It was a little bit lonely because it's the first one I've spent in 27 years without my wife. You know.
Oh.
So because she passed away in February.
Oh, poor dude.
Oh, thank you. It's to be expected. So, yeah. Maybe that's the.
Yeah, I think that's all we can do at this point.
Yeah. That's it.
How long before you get into it?
So it was once before.
I think the last one I was on was the after show after Erica.
Yeah.
Let's see.
And you're a creationist, is that right?
All right, here we go.
No?
You would probably refer to me as an evolutionist. Probably. I'm kind of undecided on certain things at this point, but I do believe in evolution.
I see.
Oh no.
Well,
so
certain
sorry.
things. Do you mean the existence of God, or Christianity is true, or something like that?
I would say I'm not sure if I'd call myself an atheist per se. I'd more say I'm just agnostic at this point, probably.
I see.
That's just my take on it.
Okay.
I kind of want to well, if you had some time right now, I want to clarify on some things that we didn't get to in our last discussion because you guys were talking about other stuff and I didn't want to interrupt.
Yeah, what's on your mind?
So I believe last discussion I mentioned this specific mutation that I think is really interesting, and I wanted to ask you about. I guess I'm trying to better understand how at least the creationist side sees information per se.
Okay. So bring it to my memory again. What mutation? What was the effect of it, or was the observed effect? You know.
Here we go. So it was a mutation in certain species of Drosophila fruit flies. In one of the enhancer regions of the gene called SOOT, which controls the number of bristles along the adult body in certain regions. This specific enhancer mutation caused an increase in the number of bristles on the forelegs, what are called the sex combs, which they use to, I guess, court the females.
Okay.
And it called. Go ahead.
Yeah, I remember responding to that query. So the creationists would view this as more bristles or less bristles. How does that move the organism towards becoming a fundamentally different type of organism, which is what evolution claims to take place so many times? And my response to that is, of course, it doesn't.
Wait, so foreskin
already exist. Having more of them or fewer of them doesn't mean the organism is moving.
Boomer made a mistake.
I'm just gonna say Nephi's been around for years. Yeah. And he knows it's a mischaracterization of evolution to say that like an organism is magically transforming into a different type of organism.
Well, of course,
what makes you think he cares about the truth or properly representing something?
Yeah, that is a problem. That is a problem. Okay, Afskin.
Yep. Or becoming a fundamentally different type of organism. The information to make bristles already exists in the genome, obviously, or it wouldn't have them. And so losing bristles, now, this, I don't know what effect this has on reproduction. I haven't looked into this. Does the loss of bristles in the reproductive area, does it affect reproduction?
It was on the legs.
That's a good question. As far as I can tell from the literature, I don't believe it does because it seems to be a derived trait in this particular species. And so that, to me at least, implies that it doesn't have very much of a detrimental effect to the population.
Okay.
And so what I didn't get to ask in the after show, or I guess at least backpedaling a little bit, asking a different question before I get to the other question that I want to ask. Would most creationists accept, like, I believe what you call genetic entropy? Could you explain that?
Yeah, I believe that they all do. They, unless they're very uninformed, I think, about their own views about creation, So yeah, it's generally accepted, and the reason it is because that's what the genetic evidence is that process is ongoing.
Okay. So you would say at least that mutations, they always are a net loss of information according to genetic entropy?
I would say yes, because mutation, the only observed effects we have that mutations have upon genetic information is that it's disruptive to it. It causes weaknesses and deformities or death.
Okay,
And
Foreskin, once again.
We don't have evidence that mutations building upon each other.
Okay, that's another one of my biggest problems with. I mean that's the fatal flaw of genetic entropy, but it's the idea that literally all mutations are deleterious,
Right.
including synonymous mutations, right? So if you have a mutation like on the third base of a codon or something that literally doesn't change the amino acid, it just changes the base that's there. You know, Sanford would say that's deleterious.
Right.
Why? I don't know.
And you're kind of getting to what I'm about to talk about.
Yeah.
Yeah. And why doesn't Leo look like my son? Why don't Leo and I look alike? Because of fucking variation. Because of
No. It's created heterozygosity. That's the only reason.
Oh yeah.
They would have trouble supporting that because they accept that we're mutating, and I think they accept that there was speciation after the ark. So obviously, my house cat does not look like a leopard.
Yeah,
they have to accept that some mutations do not result in deleterious effects on the phenotype.
I've been personally trying to push some of them towards created polyploidy because I feel like that is an excellent idea.
Oh, so you're trying to give them new ammo, so they.
I mean, it would. It's a ridiculous and absurd idea, but it would help their case with the limited amount of alleles, right?
Yeah,
if you humans are diploid, so two individuals can have a maximum of 4 alleles for any site.
So each 100 chromosomes.
Yeah, exactly. If they were just pentaploid, you've multiplied that number by now you have 10 alleles. Yeah.
Well, good idea.
Exactly.
Actually, we could get a research grant from them.
Send it out to CMI. They'd probably take it.
They'd take it.
Oh dear.
Ask him.
Yeah.
Develop new structural designs in the body plans of living things, and that would be necessary if evolution were true. That's exactly what would have had to have happened.
Okay. Pause for a sec.
And also,
you got to say.
All right. So just making a note here. This is his first definition, quote unquote, of information that has to do with structures and functions. Okay, just making a note of that.
Yeah, let's play that again.
Structural designs, weaknesses and deformities or death, and we don't have evidence that mutations building upon each other develop new structural designs in the body plans of living things, and that would be necessary if evolution were true. That's exactly what would have had to have happened and continuously happen. And also, the examples that we know of where mutations occur to information and it provides a visible effect seem to demonstrate genetic entropy. The reason being is that mutations which, in fact, let's get an example going. So creatures, it's been noticed that bacteria can develop
the citrate example.
Mutate and develop the ability to digest citrate, to utilize citrates.
Okay.
But the mutation that turns the gene on causes a negative effect. The gene gets broken. It doesn't.
No, it doesn't. Okay.
This is the gene does.
Wait, foreskin. Sorry. Okay, that's a total misrepresentation of what they found in the LTE.
Right.
What happened was the gene got duplicated. The gene that codes for an antiporter called the CITT, or the CIT transporter, basically. It transports citrate in exchange for, I think, sodium or something. I don't really remember. It doesn't matter. It got duplicated onto the neighboring gene, and it basically hitched a ride to that other gene's promoter. And normally, the gene only got activated in anaerobic environments. But since this duplicate copy was also being produced at regular rates in aerobic environments, there was always this citrate transporter. So I mean, you could say it's not really a gain of information, but saying it's a loss of information is bullshit.
Well, if you look at any physical theory of information or attempt to understand DNA as information, it sure as hell does count as a whole lot more information if the gene got duplicated. And if it basically, if it ended up next to another promoter, that adds even more information because now it's promoted in a different manner. So the cell is doing something it was not doing before, and it didn't lose anything. But I
think their argument is that it's losing a step of regulation, essentially. Like it can no longer regulate which aerobic conditions it's producing the antiporter.
Yeah, and the bacteria went on happily multiplying without that.
Yeah. Of course, though, I'm obviously, steelmanning NephilimFree's argument. He probably does not know what an antiporter is, if I had to guess.
Yeah.
Or like he doesn't know like the specific situation.
Well, yeah, listen to what he says next.
Default. So mutations, we don't have any evidence of mutation that's strictly beneficial. Mutations that seem to provide some kind of benefit at the same time seem to provide cause a negative effect.
No.
So a gene gets mutated and it no longer is able to do one thing, although it may provide the ability to do another.
So he thinks a gene got mutated. And
yeah, exactly. That's not what happened.
So he doesn't know what happened.
Okay, and even then, he said that all mutations are deleterious. And like I said, that's one of my biggest pet peeves. Because all mutations are context dependent. Literally all of them. You can make the argument that it's a loss of information, right? Like the most common one I hear, this is the argument that Sanford makes, is it's a loss of information because you're deviating from like the designed genome, whatever that means, right? So that's why Sanford would say that synonymous mutations are deleterious. But like
that only works if
it's presupposing the question.
If design was true, right?
Yeah. But saying that all mutations are deleterious is just dumb. Like, what if your organism is brown and it's a loss of information mutation to lose like whatever pigment that produces the brown coloration. And so now this organism is white. That might be bad unless you live in the Arctic. And then it's still a beneficial mutation. You know what I mean? Beneficial mutations have to happen under the creation model. Because like you were saying, a cat and a lion to share a common ancestor had to have some sort of mutation.
Right.
And the lynx, if you talk about new structures, if you look at a lynx, which is a cat, it has those wild things coming off its ear. I guess that's just hair growing longer, they think.
Yeah.
But their ears are of a different form. All cats have different formed ears. So, but they're still ears, I guess.
Yeah.
What they'll say. So, okay. Ask again.
Yeah, ask him.
And if evolution can't proceed on a process that constantly damages existing functions, especially if they're critical to the existence of the organism. But the kinds of changes that we observe occur to organisms due to mutations don't change the structural design of the organism. That's really the bottom line. And if evolution is true, then mutations do that. They constantly build new structural designs. They constantly transform existing structural designs in the anatomy of creatures so that they provide new biomechanical functions. And we don't observe that. We don't have any evidence of that from the 90 years of studying mutations and their effects. So,
except for ApoA1 Milano, which, have you noticed that since I talked to Standing for Truth about that twice, no one has talked about that fucking gene since.
Mm-hmm.
And they used to talk about ApoA1 Milano constantly. I mean, it was one of their biggest talking points that this mutation happened and it screwed up everything else. But as it turned out, the mutation happened. It made cholesterol collection a lot better. And it also made the antioxidant function about, what was it, 6 to 20 times more effective. So they don't talk about it anymore. But that is one clear example of an actual mutation to a gene, to a protein, that caused it to dimerize and tetramerize rather than form 3s and 5s and whatever like it used to. But it ended up being a whole lot more effective at cholesterol collection.
Mm-hmm.
And they couldn't find anything wrong with the gene.
Well, and also his argument about like, well, things never affect the structure of an organism is kind of dumb, because mutations affect the structure of an organism all the time. You know,
Right. He's concentrated. As we go on here, you'll see that he's concentrating completely and solely on proteins.
Yep.
Genes.
Yeah.
yeah, but well, like so, you know, as an example, there's certain diseases and stuff that most humans wouldn't consider beneficial in any way. And obviously, you're never going to get from something like a chimp to something like a human in one generation, because
Right.
that's just not going to happen. That's a misrepresentation. But humans are essentially neotenous chimps that learned how to walk on two legs, right? That's one of the reasons our brains are so much bigger, because you know we retained the childlike characteristics of chimps just because you know we didn't express all the same protein pathways. And in humans nowadays, you know, if someone expresses childlike behavior for the rest of their lives, that's obviously a deleterious mutation, and most people consider that like some sort of deformity or something like that. But it's obviously like changing the physical structure of that child. And in the correct situation where that would be a beneficial trait, it's easy to see how that can become propagated through a population.
Yeah.
Yeah, and to add on to that, like when you look at the genome of a human and a chimp, you find that although there are differences, like you said, Speed, in the protein coding, the coding regions, you also find massive, or at least a bunch of differences in the regulatory sites, and that probably I would suspect most of the differences between a human and a chimp in terms of morphology.
And most of the genome is regulatory compared to protein coding.
Right. Exactly.
We have 1 to 2% protein coding. I think 1.3 is what I've heard, but it could be as high as 1.7. But there's about maybe another 20% that's all regulatory.
Yeah.
So that is the most likely. 1 out of 5 mutations are going to hit regulatory genes.
Yeah.
And 1 out of 100 are going to hit proteins.
The information in the genome is highly specific. It's very specified. The functions that it produces are highly specified information. And it only stands to reason that if you tinker with the letters in a book, you're corrupting the words. Eventually, the passage won't be readable anymore. So that's what we observe going on with mutations. And we don't have any evidence to the contrary that mutations develop the structural design changes necessary for organisms to morph slowly into a different type of creature. There's just no evidence in genetics for that.
What?
So we can't conclude that mutations are mechanisms of reproduction. We can only conclude that they're degradation.
He said we have no evidence.
Is he making the same argument he did in our debate that mutation is not like the single driving factor? Well, I shouldn't say single, but one of the main drivers of evolution?
He says we have no evidence.
Yeah.
Except literally we do.
Except all the evidence for what happens in the cell and in the genome is evidence for that kind of evolution. Go ahead.
I just find it funny. I always love how they forget about the other half of natural selection, like the whole selection part. Mutations create variation, and then they never discuss purifying selection. Right. And when they do, it's always to just like hand wave it.
And purifying selection happens to an unbelievable degree even before we have a pregnancy. I mean, there's a lot of eggs and cells that sperm that do not make it.
Oh, not even just that. Isn't it something like greater than 50% of all pregnancies terminate before the woman even knows she's pregnant?
Yeah, there's no sign of a pregnancy at all.
Yeah. So.
Yeah, and like people talk about mutations isolated, and they talk about natural selection isolated, and they can talk about drift isolated. Well, it's everything kind of in tandem. It's not like one's isolated from the other, and I think that's a big misconception.
That's actually very sorry. That's actually really true, and that's something I don't know. You just made me sort of realize that, because that's really funny. The only time they ever really talk about selection is when they say, "Well, selection just sorts on pre-existing diversity. It can't create any diversity." It's like, yeah, that's what the other half of evolution is.
Yes.
Doesn't SFT have a shtick about isolating mutations in different species, and they eventually like go extinct. And he uses that as evidence that genes only deteriorate.
Yeah, so his whole argument is genetic entropy,
Mm-hmm.
which is something I was talking about earlier. Genetic entropy is basically the idea that, so in population genetics, a mutation is deemed nearly neutral when selection on that mutation is too weak to really be noticed by the population. It's a fact, or it's a like function of how large the population is. I think the selection coefficient for that any given mutation has to be like 1/2NE or something like that.
Yeah.
And so the argument is, well, deleterious mutations are much more common than beneficial mutations. So these nearly neutral, slightly deleterious mutations are going to build up over time and then cause error catastrophe where the species goes extinct. It's a pretty bad argument, but that would take me going into population genetics and shit.
Yeah.
So there's the other thing is that 1 in 33 babies are born with a birth defect, and birth defects account for 1 out of 5 deaths in the first year of life. So how many of those 1 in 33? Basically, it's got to be a pretty like about 50% of them die within the first year, and that is why that is purifying selection, pure and simple. And the ones that we manage to keep alive, a lot of them don't reproduce, or they have the sense not to reproduce. They know they have a birth defect. But then for animals, the babies born with birth defects are going to have an extremely high mortality rate to the point of wiping out. And these are what we could call near neutral deleterious mutations, where they will where the organism actually gets to be born, but it doesn't really function or survive or reproduce. Yeah. And that purifying selection is a motherfucker.
Yeah, and also, I think what you said was pretty interesting because it sort of highlights the idea that humans are exceptions to the rule. Because, you can have a pretty deleterious mutation, and overall, it's like the effect on your relative fitness isn't going to be that high.
Yeah.
What is purifying selection, and how does it differ from natural selection?
Purifying negative selection.
Yeah, purifying selection basically just selects out highly deleterious mutations.
How?
It's not any different from it's a type of natural selection, essentially.
Yeah.
Natural selection is just any sort of selection, and then purifying selection is selection against specific alleles, and then you also have like forward selection, stabilizing selection, stuff like that.
Calling it negative selection and positive selection kind of appeals to me. What do you think of that, Walker? Or do you like?
It's accurate.
Yeah.
There's not really a such thing as positive selection.
It would be like if a mutation caused a higher reproductive rate.
Yeah. So when you're talking in biology, you're only ever referring to alleles as being selected against, because it's a quirk of like relative fitness. So your relative fitness is basically your absolute fitness divided by the fitness of the most fit genotype. And so the relative fitness of whatever beneficial mutation you just had would be 1, whereas the original allele would be like 0.9 or something like that. Okay. So you're selecting against the original allele.
Yes.
So it's not technically positive selection, but you can think of it that way for sure.
So the positively selected would be fitness of 1.
Yeah.
Basically.
Yeah, pretty much.
Everything else would be less. Yeah.
And everything else would just be selected against.
Yep. Makes sense.
Yeah.
Okay. For Afscan?
Yeah, sure. Afscan. Tangent, but yeah.
Respiratory. Most mutations are what we would call silent. That is, they don't produce a visible effect in the organism. Right. But that doesn't mean they're not causing damage. Just like you might be able to read the book if you.
No, it fucking does mean it's not causing damage.
That's literally the definition.
and they just slide right over that. They think that there's this very tiny bit of damage or rust.
Guys.
Yeah, go ahead.
These mutations are defined as not causing damage, but that doesn't mean that they're not causing damage.
Yeah. Okay. Yep. Okay, we're going to move on.
occasionally change a letter in it, and it may not be so visible that the book's information is being damaged, but when you can no longer read a sentence, now you've got reason to believe the evidence is that the mutations, the changes, are destroying the information in the book. Well, if the genome is highly specified information and it's being changed randomly by undirected processes, stands to reason that entropy is occurring to it. It just happens in such a small amount with each mutation that the effect is not visible in the organism as we can see.
So I guess.
Go ahead. Oh, that was you. I thought you were saying something. I'm sorry.
Well,
I kind of wanted to stop anyway. This bullshit about comparing it to a book. Now, a book has words in it, and the words have one correct spelling. Okay.
Right.
And if anything happens, like we change one of the letters, then we have a damage. But that is not how proteins and genomes work at all.
Mm-hmm.
I mean, it just isn't. There's a great deal. Well, he goes on to talk about his misunderstanding of protein folding, I think.
Yeah. Well, proteins are extremely robust.
Yes.
Like everything about the genetic code is very, very robust. But proteins, like normally, as long as you're not touching the active side of a protein, you can really do whatever the fuck you want to it.
Yeah.
Like any mutation that changes an amino acid, as long as it's not to like something super different, you're fine.
Yeah, as long as you don't end up creating another fold. And even then, if you create a slightly different shape in the substrate, well, let's wait until he starts talking about.
So I want to say really quickly that with his analogy, it seems to me he's saying that, well, you can like maybe change one letter in one word, but the story of the book is like completely unaffected by that. But if you go small enough, like if you read the sentence, the meaning of that sentence or the meaning of that word might change.
Right.
But that would, if you're gonna draw an analogy to mutations, it would be like in realize mutating to an S.
Yeah. Or changing TOO to TWO or whatever.
Mm-hmm.
Would have a different meaning because we'll pick out a different word. But okay.
So I guess a question I might ask you would be if, for example, well, we all agree that the genetic code in terms of codons transcribing into or translating into amino acids is redundant, correct? So
there is redundancy in the genome. Yeah, quite a bit of it looks like.
Okay. So I guess what I don't quite understand about the creationist position is if say you have, I don't know the specific codons per se, but if you have a silent mutation or a synonymous mutation and it causes leucine, and it most likely affects the third letter of the codon, and now it's still leucine, I'm having trouble understanding how you guys would see it as a loss or a degradation, I guess.
Okay. Well, it has to be understood first that mutations can occur which change a codon so that an amino acid is substituted with a different one. That does happen. It's rare, but it can happen. Mm-hmm. And the odds are that the amino acid
kind of rare. It's 1 out of 100, roughly.
I would say uncommon, not rare.
Yeah. Well, I mean, so non-synonymous mutations are more common than synonymous mutations, but that's assuming that any mutation is happening in a protein. And since like proteins are only 1% of your genome, it's relatively rare.
Well, Walker, I would kind of disagree. I would say synonymous mutations are more common.
Are they?
Why would you say that? For like selection rates, or
I think it's 36%. Do not change the amino acid, and something like around 30%, so 70% do change it.
Yeah, that sounds about right. Because really, you're pretty much always going to have a synonymous change that happens in the third base pair. It's fairly rare to happen in the second base of the codon, and then you're never going to have a synonymous mutation if it happens in the first one.
Yeah, I got that 30% from running a program and running it through a bunch of random mutations.
Yeah,
and
I mean that sounds about right.
And checking to see it, so it's a mathematical thing.
I mean, so synonymous mutations are going to be more likely factoring in selection, but I was just talking about like de novo mutations.
So about 0.8% would actually change an amino acid, and because proteins run about 40% redundancy, we end up with about 0.4%, even less, maybe. I don't know. Probably less. Depends on, I don't know. Anyway, ready to go on?
Yeah.
Sure. Well, we'll see what he's about to do.
Which
Yep.
gets substituted, is not going to provide the exact same fold as the original one. There are occasions, which is rare, when one amino acid can perform the exact same fold.
I just want to point out that no, it takes a whole lot to change the fold.
Yeah.
Unless you get an unlucky proline substitution, maybe, and that'll put a little kink at an alpha helix or something like that. But changing the fold he implies that if you change the amino acid, it's going to change the fold, and he believes that, and he kind of doubles down on it, I think.
Yeah. There's so many like hydrophobic amino acids that look the exact same, except one will just have like an extra carbon group on it.
Yeah.
That's obviously, switching one out for the other generally isn't going to do anything.
Yeah, I've got some really intense books on alpha folding, and it basically, they've done some really intense studies on amino acids and what does change things.
Yeah,
but I haven't had a chance to dig into them yet.
Little fucking nerd.
Well, also, I just wanted to point out, notice how I asked about synonymous mutations, and he started talking about non-synonymous mutations.
He just completely blew that part off. So
yeah.
Okay.
If for the protein chain, as the one it's substituted for, that's possible. That's uncommon. It's very uncommon. It's rare. Typically, a mutation which causes a change in amino acids so that one is substituted for another results in an amino acid being substituted which does not provide exactly the same fold as the prior one, the original. When that happens, the protein has been degraded because its structural design, the shape of it will change even depending on which amino acid in the chain we're talking about. The results of that can be either completely disastrous or minuscule in effect. Right?
Okay, so that's bullshit because there is no reason why it cannot be slightly better fit than a slightly worse fit. And they will say it was designed for perfect fit, right? And they will say that any change is moving away from that perfect fit. As it turns out, a lot of changes don't change anything about the fit. And furthermore, there's I lost my train of thought. There's what the hell is it gonna? Oh, there's no reason why a mutation, like for substrate binding, there's no reason why a mutation couldn't as likely increase the substrate binding as decrease it.
Yeah.
Really reason.
That's what I was thinking. So what would Nephi say about like a back mutation where you're mutating back to the ancestral genotype?
That would be a gain in information.
Yeah, but that should objectively be a gain of information, right?
According to his definition.
Yeah.
Of perfection.
Yeah.
So,
I think he would probably just deny they exist.
Go ahead.
Oh, I was just saying he probably would deny that they exist or say that they're so rare that it's not a factor. But, when you have a reasonably large population, back mutations and forward mutations are in equilibrium. So
I would say they're rare. I wouldn't say that they're irrelevant. Or I wouldn't say that they're irrelevant because, I agreed with him here that there, in some environments, there are more ways to make an organism less efficient or worse than it to make it better. I agree with that, but that doesn't mean that the beneficial mutations don't exist at all.
Yeah, well, so the thing is with back mutations is they're comparatively rare, but when you have a large enough population size, eventually there's going to be only so many forward mutations that can happen, and then the back mutations start becoming more frequently, and then it just reaches an equilibrium point, right, where forward mutations are happening and back mutations are happening. I don't remember the actual rates and stuff, but it happens at a relatively large frequency when the population's big enough, essentially. Just because there's 7 billion humans and there's 6.4 billion base pairs. So any mutation that could happen is currently in somebody that's out there living today.
Speed.
Yeah.
Question: do you have Dan's email?
I do.
I can send
Oh,
it.
really?
Yeah.
Yeah. Do you see if he wants to join? I don't know. Might add to the conversation.
Yeah, I asked him.
normally
I didn't know how long's he been in the chat, and I've been missing everything.
I think that was his first one.
Okay, so we don't have a whole lot of chat yet.
Yeah.
You guys aren't very chatty tonight. Let's have some chatty.
Yeah.
I could contribute. I just popped in. What are we talking about? We're going over a video by NephilimFree where he talks about genetic entry, and Andrew is trying to get out of him what his definition of information is, and he kind of leaks it, right?
Yeah, basically, creation myths, Dr. Dan. I'm trying to get him to answer what we agreed on to call the bristle question, if you remember what we talked about earlier.
Yeah, it's about the bristle question. I should have named it the bristle question instead of NephilimFree in our sites.
Oh, yeah. Oh.
Anyway, you sent him an invite, Walker?
I have not, but I can.
Oh, I'd like you to.
Yeah.
Okay.
I guess we'll keep going and see if he joins then, Speed.
I know it's your day off because you're ill, but okay, we'll go on.
A very complex long chain of amino acids, and they fold. Which amino acid is substituted will have a greater effect on the outcome, the final shape of the protein, than others. You understand what I'm saying?
Yeah.
You understand that? Okay.
Yeah.
Right. You sound like somebody who has enough understanding to know what I'm talking about. I think you understand. So let's say if the fifth amino acid is substituted with it wrong.
Professor Nephi over here, I guess.
That might have a different effect than, I'm saying this for the audience's sake, than the 20th one or the 200th one. The 200th amino acid substitution may have a lesser effect on the outcome, the final shape of the protein, than the fifth one, right? May not. It depends on the exact amino acid. It might even depend on whether or not that particular amino acid is on the outside or the inside of the protein.
That's correct.
Finish folding, right? Because an amino acid which ends up on the outside surface of the protein will have a change to one of its folds, will have a greater effect, probably, than one that's somewhere in the interior of the molecule, right? But we don't know that for sure. That's just a speculation, but it's probably true in a general sense. So because those are the places where that protein bonds with other proteins to provide its biomechanical function. If you change the way it locks up and links with other proteins, now you've had there's going to be some kind of effect upon functionality. Right.
Right.
So,
I would agree that if you change, for instance, the active site of an enzyme, for instance, that's going to change how it binds with the substrates. I would agree with that.
Exactly. Right. So, but the thing is, when medical science demonstrates, enough studies have been done, quite a few, many, in fact, to the effects of mutation upon protein codes. And amino acid substitutions have been studied a number of times because this is the cause of so many diseases. Naturally, medical researchers are interested in knowing all they can about changes to protein sequences because they are known to be the cause of the preponderance of human diseases is a mutated protein. That's the single greatest cause of human disease is either...
No, it ain't. Well, it is in the somatic line, but well, maybe not even then, but I don't know that mutations are the greatest cause of human disease. Birth defects are certainly not the greatest cause of human disease.
Right.
And he's being disingenuous about we've studied protein mutations and we've found out they're bad, and then he kind of slides on him. Of course we study mutations. That's what the hell it's all about, basically. Right. And he kind of implies that they're studying them because they're finding that they're all deleterious in some manner or whatever. So he's trying to load up the question here again. So.
Yeah. All right, let's see what he says.
The ability to produce enough of a given protein, or on a rare occasion of producing too much of a particular protein, or a change to the shape of that given protein, and that's which results in a change in its efficiency or function. Right? So medical science is very interested in mutations of protein codes, and it's been studied a lot. All kinds of universities have studied that, and the overwhelming evidence from their studies is that mutations cause a negative effect in general to the functionality of proteins. When an amino acid substitution occurs, the result is typically, not every time, typically, the vast majority of the time, results in a protein which has reduced efficiency.
Bullshit.
Which is one of the causes of hypoglycemia, for example.
What the fuck does that have?
Or complete dysfunction. The protein is not functional anymore at all, and now the person has a disease, a severe one possibly. So that's all we observe the effects to mutation. That's what we observe the effects are when amino acids are substituted. Is disease is the typical result. So either mild or severe, one of the either one.
Can you pause for 1 sec? You might want to skip ahead a little bit.
Now I'm confused. Okay.
Yeah, you might want to or
skip ahead.
I got 2 phone calls in the middle, so maybe skip ahead a couple minutes.
Okay, I'll try.
Okay, sure.
I thought you were actually telling me to pause this video, but you were on that video, and that.
Discussion.
Yeah. Next we start talking about the difference between gain of function mutations and gain of information mutations.
Okay.
Fun.
Okay.
We also been
function, and that the protein has function or not function cause a loss of feature of function. Yes, that's known to be true.
Did I go too far?
Okay.
No, I think you're good.
Gain of function doesn't mean necessarily doesn't mean that the protein has function or not function. It might mean gain in its ability to bind to a particular enzyme or something.
Sure.
But it doesn't mean that a new function has arisen. Okay, so people misunderstand that term when it comes to mutations in proteins. They think gain of function. Even many evolutionists who don't know better think, oh, well, since it's called a gain of function mutation, that means a new function has been gained. No, it doesn't. That's not what that means. I think you understand that. But a lot of layperson evolutionists don't. They just hear the term gain of function and think, oh, see, look, evolution gains functions. That's not what that means.
Sure. Sorry to interrupt the phone call. Sorry, I'm getting an actual phone call.
Okay. Yeah, go ahead, take your call.
Looks like we caught the phone call anyways.
Yeah. Oh well, sorry about that. It was a spam call and then it was an actual phone call.
Yeah.
Okay.
A lot of phone calls.
That happened to me today.
Yeah, in the middle of the live stream too.
Yeah.
I think it started again at about 13.
Hold on. Number one, what are you and Lena going back and forth about in the chat, Speed?
How many beneficial mutations there actually are, and
are you speaking generally?
I disagree with the kind of standard talking point that beneficial mutations are rare. And
are you speaking generally though about just mutations in general across all life?
Right.
Yeah. The paper she's citing is about specifically asexual populations.
Mm-hmm. Which means it's subject to like Mueller's ratchet and shit.
Yeah, but so like what she's citing doesn't actually, I would assume, does not actually apply universally to all forms of life.
Okay. Yeah, well, when you add in recombination, you can fix a lot of the problems that asexual reproducing organisms would have. So that's the whole thing behind like Mueller's ratchet. Mueller's ratchet was basically Mueller predicting like asexual populations should experience air catastrophe, and that's why diploid like recombining sexual organisms have like a selective advantage.
The number 2 thing I wanted to ask is we always talk about amino acids. Why don't we ever talk about aniso acids?
That was good.
Mm-hmm.
I had to.
Or amino acids.
Oh god, I forgot what I was saying.
Okay, so I just have a difference what they call.
And also, that's where I was originally going, but my train of thought's kind of fucked up right now.
I would probably.
Makes 2 of us.
What happened?
I would probably
make 2 of us.
Okay.
Oh well, I'm just saying, like, if you have any beneficial mutations, then you're going to have selection, and then you have evolution. So it doesn't matter what the rate of beneficial mutations are.
Okay, so now Scott has a good question. Were Nephi's words really gain in function mutations are not really gains in function? Like, so is Nephi correct there that it is not actually a gain in function, despite it being called a gain in
function?
Yeah,
my intuition tells me he's wrong, and my experience would tell me that he's wrong, but I don't know enough on the subject to actually say that he is wrong.
I think I figured that much out when I was about 4 years old. That when Mom said "don't do that," she didn't mean do that.
Yeah.
I mean, when Mom said "don't do that," it actually meant "don't do that." I don't know.
Yeah, like gain of function / loss of function mutations, as far as I remember, has to do with the
function or activity of the protein, not necessarily how or I guess how well it does it, not necessarily if it does something new. I think that's the definition in the literature.
Yeah. Oh, let's see.
Let's hear.
You want to go further?
Let's touch. Sure. What?
Want to go further?
Yeah. As long as we get to skip Andrew's call.
Okay. So wait, but I did really quick. The whole point of this discussion was to figure out what Nephi's definition of information is.
Yeah,
creationists don't have one.
Well, he kind of leaks what he's thinking, but he pretty much explains that also they don't really have one, right?
Yeah. Well, that's because they think that information is non-physical. There isn't really something there to be defined in their worldview, which is why they struggle to define what information is.
Right. Yeah, but like the point, the bristle question isn't to get them to I guess if they want to define it, sure, but more so the point is to get them to quantify it, and if they can't, to show them or to show the audience that they don't have a good definition. That's not quantifiable, basically.
Which is inherent to any definition of information that claims that it is an immaterial substrate, whatever that means.
Exactly.
Here's my challenge for the creationists.
Well, I mean, it's honestly not that bad. It's like basically a water solution with probably a bit of NaCl buffer, which is just table salt, some glucose, but like very small amounts of glucose, and then a relatively high citrate buffer as well. So, yeah, you definitely could. There's nothing stopping you.
It is 7
from drinking it. Yeah, except that we have a bunch of bacteria that have been mutated for 60,000 generations, and we have no idea how many of those mutations could be of the nature that if it got into your gut, it ended up being a really, really good fit for your gut. We don't know that. Because if you took any of those, I mean, he's got wildly mutating bacteria, mutating at a higher rate than usual. Some strains are mutating faster than others. And if you drank those or put those in a natural environment, hi, Leo.
It's 7:55. I'm going to take off. Demeter is in the side chat. If you want to stick him in, I don't know if Dan plans on coming in, but both of them should.
Okay, I'll put it out in the stream here. So
all right, we want to take it off, guys. Gotta go watch a treasure hunt.
Okay, we want to keep this mild. So
yeah,
because we do want to get through the video.
Yeah. So see if Dr. Dan wants to join, and if not, I guess it'll just be the three of us. Would you guys agree?
Yeah. Okay. So are we on a phone call?
Yeah. It's for it's for a little bit. Action is not equivalent to information. Staying. That's okay.
Sure.
So I guess what you're saying. From what I got, you're saying function is not equivalent to information. Is that correct?
That's correct.
What?
The ability of a protein to bond with another, that it in a way that it wasn't designed to, or that doesn't provide an actual new biological function, we wouldn't consider that to be a product of new information. Because is it functional? Gain of function in genetics terminology doesn't mean a new biological function. Sure. It just means a new molecular function has occurred. That's all. It's not the same thing. And but we don't have any reason to believe that mutations build new genetic information. They code for functional things. That's the thing. And if evolution were true, it would have to be so. So on top of that, because information in the genome is complex and specified, its specificity, then we for evolution to be true, it would have to do things that we don't observe mutations to do. We just don't have the physical evidence that mutations do what evolutionists claim they do. The specificity is a product of mind because it pertains to things that are down the road that require function. They have meaningful, and so if they're meaningful, then there was a mind involved. That's why we consider it information. For example,
okay. So Did he like contradict himself a couple of times in there?
With regards to what?
Well, what the hell was he saying? Okay, it's not information. Okay, function is not information because because a mutation would cause harm and it would decrease the function. That's why a function is. I don't understand what the fuck he was saying.
Yeah, it kind of confused me a little bit.
Confused me a lot. That's why I would like a transcript of that part, because I think he was just blowing steam at that point because he knew he was a little bit underwater. That you push his head down and he just started babbling, hoping that something would stick, I think is what he's doing.
Yeah.
Should we try listening to that again?
Yeah, sure.
Information that sequence
beyond this, we're going to start talking about developmental changes after we're done
information in the genome
function.
Is it functional?
Okay, so I think right here,
that it wasn't designed to, or that doesn't provide an actual.
No, it was actually how far back was it? Okay.
All right, try there.
Okay, you're still on the phone call, so let's listen carefully. I try to parse this.
Okay.
Did I go back too far? Am I running?
Yeah, it's running. I think I'm in my phone call.
You better get it.
Okay.
Yeah, I'm here.
Okay, sorry about that. I am going to have to leave shortly in about 10-15 minutes, so if you want to move more quickly with our discussion, if that's okay.
Sure.
So I guess what you're saying, from what I got, you're saying function is not equivalent to information. Is that correct?
That's correct. The ability of a protein to bond with another that it in a way that it wasn't designed to, or that doesn't provide an actual new biological function. We wouldn't.
But you were talking about a gain in function, right?
Yeah, I was talking about are gain of function mutations the same as what he would call gain of information mutations, whatever that means.
Right, and then he started talking about loss of function.
Right.
So that's where it got confusing.
Yeah.
Because he just told
like if we were just talking about enzymes, I was talking about is like the activity of the enzyme different than the information, the function of it.
Okay, we got Otangelo Grasshole and John.
I'm very glad this is an account.
Okay. Okay. Let's see what else he has.
All right.
Consider that to be a part of new information, because is it functional? Gain of function in genetics terminology doesn't mean a new biological function. It just means
what?
Gain of function doesn't mean a gain of function.
Yeah, that's basically.
Wait, where's he going with that?
I don't know. Okay, so gain of function does not mean a gain of biological function. Wonderful, Neff.
Yes, of course.
Into new molecular function, it has occurred.
Oh, that's molecular function.
Okay, so
not biological function, but molecular function.
I think.
Oh no, I was just saying. Okay, good.
Oh, I think what he's talking about here is he's not looking for like differences in the function of a protein. He's looking for a new function, like a brand new function.
Oh, so he's looking. Okay, so this is where he starts to home in on his real definition of information, which is that it would make a new leg, a new kind of leg, not a longer leg, but a brand new leg, right?
Right. Yeah, and that's a mischaracterization of evolution.
Right. He's going back to his first definition, which is structure and function.
Yeah. Lena and Dr. Dana, I define information as any change, basically. So if you had an AAA string, it would have less information than a GCTA because you could, basically represent it more simply. But information is simply a change in the genetic sequence. And if any mutation is new information, because it is different. And they want to say the only new information is information that is beneficial, but not only that, they want to say that it's new information that creates a new kind of structure in the phenotype. And then Nephilim is never very good at even defining that, because if like your femur increased in length, for a species, if they had basically longer forelegs, he would not consider that new information. He only considers new information growing a new bone or something.
Yeah,
right. He's in this case considering like new proteins, for instance, as new information, whatever new in this case means.
Yeah.
Wait, so you just define information as like a change in, or like less repeated bases?
Gain of function doesn't mean it's selected for. Let's see. Gain of function just means doing something new, a new activity, like a protein with a new substrate or something. I think he's saying gain of function doesn't mean it's selected for. Not sure what that means.
I mean, that's true.
Okay. Yeah, no, I don't think I could get creationists to endorse that because they don't know what the hell they're talking about.
Did you send a link to Dr. Dan Speed?
I put a link in the chat, and Dr. Dan he's been dissing us. He's not coming in. I'll try it again. I'll put some more bait out there for him. Okay, I just dropped the bait in the water. Anyway, Lena is quite thoughtful. I like Lena.
Yeah.
Okay, so go on. Let's try to parse some more of this.
Sure.
Same thing, and but we don't have any reason to believe that mutations build new genetic information.
What? Okay,
you're going to hear a lot of that.
Yeah, we don't have. Okay, in other words, we have no evidence that new mutation can change function, even though we know that mutations that are not neutral change function.
Right.
One way or the other, up or down, but there's no reason to go up or down. They think up is a little rare, but I kind of have my own opinions on some of that. I think a lot of the neutral mutations that are happening now are beneficial in 1 to 1,000 to generations going forward. They're going to fit into new biochemical pathways. And there's going to be change in the future or new developmental pathways, and they're going to make a difference. So,
yeah.
Okay, Lena, this definition of information would be any change from one thing to another. Like if you have a G and it changes to an A, basically by mutation, that would be new information. That is different information or new information. Information does not have a direction.
Right. It's like if you were to take a screenshot basically of the genome, and you took another screenshot in about a few generations, there would be differences, and that means that it wasn't the same as before. It's new information.
That's new information. Information is any change, basically.
Right.
Or whatever the structure of the genome is one set of information, and new information would be making any change to that at all.
Yeah.
Which is the commonly accepted definition of information.
Right.
Except, for Shannon information, which is a little spookier. Shannon information would say that AAA is, I don't know. I don't want to get into that because I don't understand it well enough. Okay.
That's the thing. And if evolution were true, it would have to be so. So on top of that, because information in the genome is complex and specified, its specificity, then we for evolution to be true, it would have to do things that we don't observe mutations to do. We just don't have the physical evidence that mutations do what evolutionists claim they do.
And of course, the ApoA1 Milano is a refutation of what he just said. And I'm sure there are hundreds and thousands of other places where we've seen mutations, and there are millions and billions where we haven't been able to observe them because we have trouble observing genomes. It takes time. How many genomes have we observed so far? Walker, do you know how many how many species?
Like how many genomes are sequenced?
Yeah.
Well, I mean, there's a lot sequenced, but there's a difference between saying that like these genomes are sequenced and that these populations are observed. You know what I mean?
Yeah.
Because like a lot of them is, we sequence just one genome and we're not seeing like how it's changing or how evolution is affecting those populations.
Yeah, we're not out there in the woods following a set of beetles around for 1,000 years, collecting their genome every 3 weeks.
Right.
And like if we did that.
Right, like a single genetic mutation can take years to study.
Yeah.
Well, and also like most mutations take a long time to fix, which I guess is what Speed was saying. But like to have any measurable effect on like fixation rates or like measurable, like it's extremely hard to show something having a substantial change in allele frequency unless it's a super tightly controlled study. Because if you're measuring, so you have 1,000 beetles out of a population of 1 million, and you've been studying those beetles for even 1,000 years. If you're only using a sample size of 1,000, it's kind of hard to determine like what the allele frequencies are.
Yeah. Hi, Demeter.
Hello, guys.
Okay, so let's get through.
We can sequence entire human genome just for 3 days.
Yeah.
Right now.
Well, so humans are obviously the best studied animal on Earth.
Yeah.
I probably have to show you how to browse the genome. So
but it takes us 20 years for a generation. So in order to look at changes in humans, it's difficult, but we could conceivably get a Drosophila. I think we've got a lot of sequence copies of Drosophila, so we could probably start to look for stuff.
Well, even humans. I mean, there was just 10 years ago so-called 1,000 human genome.
Yeah.
Also, right now there is hundreds of thousands probably sequenced. So they, of course, not displayed all of them, but sometimes you can see the difference. So that's why you have different builds in the genome. So if you go in the USCIS browser, for example, and this is my actual working horse browser for the genome, you can see that you have you have many different builds, meaning sequencing, revising. And the good thing about that is you have also different tracks laid on this. So you can virtually directly you can compare the differences between the different genome to some extent. You can see lots of SNPs also, which are in fact in the past single nucleotide changes, mutations. So it's all called SNPs. So we know about genome a lot.
So each one of those tracks are a different allele for genes?
No, you tracks for different features to lay over the genome that you can look for. For example, if you have a region of the genome, you can see for this particular genome different things. For example, you can see what kind of transcription factors has been found, what kind of conservative regions you have. You have conservative, what kind of repeats is in this fraction, and many other features. The CPG islands, and so many things. So, and this is the problem sometimes, because if you're working actually with specific build for human genome, for example, or mouse genome, for this particular build, they're accumulated during the time from the different research, you can see many features. But now it's a pure new build. And all of these tracks from the previous build of the genome have to be simply recoordinated to the new build. And that takes a time.
Yeah.
So
you should show us sometime.
Yeah, I have to show you sometime. Maybe tomorrow.
Yeah, that would be good. Maybe at 2:00. Maybe we could do that for the weather report.
Sure, yeah.
Okay.
You just need computer and internet.
Yeah. Oh, I'm going to have to go get one of those at Best Buy. Okay.
The specificity is a product of mind because it pertains to things that are down the road that require function and they have meaningful. And so if they're meaningful, then there was a mind involved. That's why we consider it information. For example, the information that's sequence of a protein. What I mean.
A lot of question begging.
Well, yeah, and also, I need to get something to drink. But he's basically ignoring that exaptation is a thing, and how mutations that are neutral for a very long time can have an effect on fitness when a different selection pressure is applied. Obviously flight feathers were an exaptation from just thermal regulation that dinosaurs used. And there's a bunch of other examples. Duplications are pretty much always exaptations, because you'll have a duplication of a gene and then there's a couple of mutations there and then boom, you have a new function.
I got a feeling Otangelo Grasshole has a link for us here that we should all check out.
Okay.
Hang on a second. I gotta ask a friend of mine a question. Okay. Ready?
Yep.
Amino acids in what order produce the knowledge that's being conveyed by the information is what the final structural shape will be of the protein. You see, that's the knowledge that was recorded as a sequence of amino acids. You see, that would constitute information.
So you would say information in this sense is a emergent phenomenon?
No, I would say information is not an emergent phenomenon.
Okay.
Information is knowledge conveyed from a sender to a receiver using a language agreed upon by both parties.
Here it is.
And so we don't have any reason to believe that mutations can create that.
Okay. So I guess going back to my original question, if that's okay, so you were talking about examples of missense mutations where an amino acid is changed. I guess what I'm interested in is, does the same information apply if it's a synonymous mutation, a silent mutation, from one amino acid to the same amino acid, but the letters are switched?
Yeah. Well, then it would just be a happenstance change of the amino acids. You wouldn't be writing the information. The information is already written. It would be substituting a word in a sentence with a letter in a word that provides a French spelling of a word instead of an English one. Or if it was the same amino acid, it would just be replacing it with the same information.
Okay. So a neutral, a silent mutation does not constitute a loss of information. Is that what you're saying?
No, the information lost would be the original amino acid. You do that enough times and you've corrupted the information.
Wait,
if you're copying it over you're replacing with the same information.
Foreskin?
There's no
meaning. Sorry, I was looking at T-Jump's email. So.
Does he literally not know what a synonymous mutation is?
Based on what we were talking about, I don't think so.
Yeah,
because he was saying well, no, you're still having a change of amino acids, and it's no, that's the opposite of what a synonymous mutation is.
Right, I keep talking about the amino acid being the same for the mutation, and he keeps going back to the amino acid changing. That's not what I'm talking about.
I would not be surprised if his understanding of translation is that each base is an amino acid or something. Each base, instead of a codon system, every single base has some sort of function in the final resulting protein.
Right.
Yeah, and he's so he's kind of showing what creationists are kind of thinking, that God wrote this information and he carefully planned it to create these critters with 4 legs and fingers and whatever. Are we proceeding?
Oh yeah.
Yeah, sure.
You gotta
I'm gonna get some water though. I'll be right back.
Okay.
All right.
I could use something. I could use a drink.
Information. If you replace it with something else that provides the same function, you've damaged the original information. You keep playing that game very long, and you don't have a protein that functions at all. And it wouldn't take much.
I guess the terminology that we might differ on is perhaps the difference between a change of information and a change in the sequence. Would you agree?
I don't know that I would agree with that. I don't see reason to believe that mutations build information at all. There's just no reason to believe that. The only evidence we have is that mutations damage the information. That's why mutations to protein codes is the single greatest cause of human disease. If evolution were true, it's the origin and diversification of proteins that result in fundamentally different types of organisms.
The whole brain trust makes a big thing of this. We're finding 25,000 mutations a year that are causing birth defects or something like that. There are 133,000 kids born a year, I guess, with birth defects in the U.S., and they are classifying more and more. But it isn't like these just popped up. I mean, these some of these things have been with us for a long time, and they kind of conflate all that, and they keep making this big thing that we are so mutant. I mean, there's so many problems, and the truth of the matter is, like for autism, we're getting far better and far tighter at diagnosing autism. And before, those kids never got diagnosed. They were just not looked at.
Right.
The rates of autism are going up in basically white middle class much faster than any other group, and there's a reason for that.
Right.
It isn't that they're mutating more. So
right.
I don't know. Anyway,
so
the proteins in human beings.
Go ahead, Demeter.
So 2 things. First, back to the autism. I'm not in this research. I don't know the details a lot. However, whatever I just know so far from 1 or 2 papers maybe, is that we are talking for mutations in a different genes, but we're talking about, let me say, something between 50 or 80, let's say 100 genes.
Wow.
But those genes could be mutated in different ways, differently, but those genes are responsible for the final stages of brain development. So you can imagine a completely normal person. Of course, he will develop childhood, but at some point, those genes have to be triggered and to do the job. So at the final stages of the brain development, now kind of disrupted, and let me say the neurons, for example, don't know the details here, but let me say some neurons cannot make specific connections because some of the proteins are damaged. So we're talking for real DNA damage, so mutations in approximately 80 genes or something like that. But you can also see something else. Yes, autism is indeed an awful disease and could vary, of course, from very, very damaged brain to much lighter, not so damaged situation. But sometimes that can trigger also a specific function in the brain which is exaggerated. Meaning some autics actually function better in some specific things. Let me say, it's happened sometimes that brilliant mathematicians could be autisms. or something, or just some mental ability. It's rare, but sometimes it happens. So see how the evolution works. I mean, imagine just the lucky mutations where the brain is just pushed in something better to do, and that it's kind of genius luck. So
yeah, back in the 60s, 70s, 80s, autism was defined. If they said somebody had autism, they were talking about almost completely loss of function for the child, and they called it autism. And now there's a spectrum of autism that includes some highly functioning individuals. So I mean, saying that more and more are diagnosed now isn't saying a lot. I don't think.
Yeah.
I don't know what. There's no indication that the rates are actually going up, basically, any more than our population is. So I don't know.
So the second thing that I want to mention. So I was listening to NephilimFree almost 40 minutes, right? So lots of BS was happened. Well, I always used to say, in the field of mutations or all of this exchange, whatever could happen will happen. It is capable to happen. So usually, mutation, first of all, to judge the mutations, you have to know about the structure and how the actual function works. Starting from the mutation, you can have, of course, everybody knows that. I'm just repeating this. You may have point mutation, just a single nucleotide mutation. You have deletion, insertion, a bigger junction, bigger fragments of DNA could be deleted, could be inserted, could be reversed, and also could be just duplicated. And all of that happens. And even more, there is a polymerase in existence, which is linked to the DNA repair, which polymerase is capable to add nucleotides without template. Can you imagine? So there is a polymerase that can template, just polymerase random nucleotides and make completely new string of DNA. Of course, this new string is just designed, meaning it is function. I mean, function here, not intelligently designed. It is designed just to fill up the gaps where you have two joins. So during the DNA repair, there is a polymerase which can just add nucleotides to have a junction, to make a junction for this. So here we go. You just generate new DNA. During the DNA replication, for example, it's very frequently when you watch the genome, you see how repeats are going on. So you have numerous hundreds of thousands of repeats. Let me say GC, hundreds of times, or CC, or A. And this is effects of slippage. Sometimes during the replication, you can have an effect of slippage where just one side of the DNA is flipped and the other side is continue to repeat the same sequence again, and after that is just stretched out. And this slippage could happen in negative, meaning removing some of the DNA or adding some of the DNA. Many mutations happen during the DNA repair, for example. And also, most of mutations actually are fixed with the repair mechanism. So that's one point. So you can have all kinds of different mutations in all possible variants. But in order to function something as a new function could happen just with rearrangements. I mean, that's obvious. You can rearrange some domains on a DNA level, of course, and that will be transcribed in a new protein. So one of the, we see that in front of our eyes when we are studying cancer as a mechanism. very frequently, actually, cancer came to the point of genome instability situation. Just because there is a non-well functional DNA repair mechanisms, the genome became highly unstable and started to rearrange different regions and to delete some region, to amplify another regions. And here we go. We have completely well established and studied by us mechanisms how the mutations come and how new function can come. So to me, this is just overall, I don't know what Nephilim tried to look smart, use some terminology which I never heard about this terminology, although I'm 20, 30 years in the field. What kind of terminology he is using. But once you go to the details, to the actual what is happening, you don't need of this fancy terminology. You just see what actually happened. That's how it is.
Well, the interesting thing about cancer is that it's actually functional. I mean,
it is functional.
Yeah. And it's from mutations, and it ends up creating cells that divide like crazy.
Exactly. So there's two ways to do cancer. One way, because there's in respect to cancer, there's two types of genes. One type is called oncogenes, and the other type is called tumor suppressor genes. So if you somehow eliminate the function, that doesn't mean delete, or just you can just silent tumor suppressor function. In that case, the other will take over, which will be the oncogenes. Also could happen, the oncogenes can be amplified. For example, MYC gene is very famous oncogene, which once it's amplified, unfortunately, in some children's cancers, unfortunately, MYC is heavily amplified, like thousands of times, and produces super heavy and super aggressive cancers.
Yeah.
But that's a different topic. So.
Yeah, the new thing that they're saying in all the creationist channels is that the experts aren't keeping up to the literature. They're writing the fucking literature.
Oh, bad scientists. Awful.
Yeah, they're writing it, but they're not reading what they wrote. Is that what they're saying? I don't know. It's just like, and yeah. Oh my God. That is egregious. Anyway, okay. I love this Otangelo Grasshole. He believes he come from monkey and he related to tomato and puppy come out of pine cone. Who the hell is that? Does anybody know? Maybe we don't want to give it away. I thought it was Ian, but he hasn't responded to me.
Yeah, I was about to say I have like two or three guesses.
Yeah, Ian was one of them. John Peterson was another one.
Yeah, those are my top two as well.
Yeah. Okay. Ian has not responded to me, so either he's hiding out there under the palm trees or he's on vacation, I think. Okay.
Yeah.
I'll move on.
All right.
In chimpanzees are all 80% of them are different by one or more amino acids. Oftentimes 3, but sometimes as little as 1.
Wait, foreskin.
Okay, so
did he say chimpanzees are different? Like all chimpanzees are different by 1 amino acid. Sometimes it's as much as 3.
Yeah, something. Well, he's talking about each gene. 80%.
That would make more sense than each chimpanzee only differs by 1 to 3 amino acids.
Okay, but like 80% of my genes may be different from yours by 1 amino acid.
Yeah.
I don't.
Very likely so. And also, chimpanzees have higher divergence estimates than us.
Yeah.
Like the last common ancestor of all chimpanzees was like almost 1 million years ago, I think. And then the last common ancestor of all bonobos was it was still precedes us. It was like 500,000 years.
Yeah. So, Demeter, do you know what the number of, like, if we took all of my genes and all of your genes and compared them, what percentage of genes would we differ by, me and you?
Well, I don't know percentage, but you have to know the principle of the game. So you, as a genome, you and me, we have completely identical lots of genes.
Okay,
I mean,
so completely.
everything that it's very important, usually it's very conservative as well.
Right.
There is regions in the DNA which is between the genes, which is very highly variable.
Yeah.
And you can find in those regions individual difference, meaning you can use it in forensic science. Usually, those kinds of regions are PCR'd by forensic scientists just to find who actually is responsible for the crime. But this is a regions which is satellite. Usually, it's satellite repeats or some kind of repeats, which is highly variable.
Okay.
And also, there are lots of SNPs, SNPs. Also, we can say.
Yeah, that's what I'm wondering. Between the two of us, how many?
I will show you tomorrow. I will show you tomorrow the SNPs, for example.
Okay.
But you will see that lots of SNPs are accumulated between the exons.
Yeah.
And when you go in the exon area, there is SNPs, of course, but not that much.
Not that much.
That many. So it is.
The other question I had is on introns. How much of the intron is variable and how much is highly conserved?
Of course, introns are much more variable than the exons. I mean, that is obvious, and you can visual. I mean, you can see that.
Okay.
There is no doubt. I mean, what kind of? I mean, this NephilimFree, what he's talking about? He's trying to make a kind of examples by one or two amino acids. What does it mean? Nothing. I mean, there's so many variations. You have to just go and look what he's talking about. I mean, I can show you the genome and you can see all of the regions which is between the exons, and if you go between the genes, it's so variable. It's, of course, there's to some the very close species, even very close human beings. I mean, very close individuals are very alike in these regions, of course. Yeah, you can go and just.
Okay, we're going to go on a little bit.
Sure.
Right.
Evolution were true, then proteins evolve constantly because the batch of proteins that exist in the cells of a sea urchin are going to be very different from the batch of proteins that exist in a human cell, right? There's going to be a lot of differences because they're different creatures.
I will say yes and no.
Evolution allegedly.
Yes and no. Yeah, some are.
Yes and no.
Yeah.
I mean, there are lots of. I can guarantee you with the urge. So you have lots of absolutely almost identical genes. Especially genes related to the translation, transcription, replication, some genes connected with the metabolism and so on. They're very alike. They're sometimes, because, for example, sometimes from my projects, I was trying to figure it out how to distinct mouse gene from human gene. And that happens at least once happened in my projects before. And I can tell you directly from experience, it depends on the gene, but very frequently I'm puzzled how to find this particular spot, which is so different, to be sure that by PCR I can catch exactly only the human part. So
yeah, like I looked at one gene, that DDX11, and the mouse was 52% different from humans and chimpanzees. Humans and chimpanzees were about 7% different, and that was amino acid substitutions.
Yeah.
So the actual protein was 52% different in the mouse, and apparently did exactly the same thing. So, anyway,
okay, let him go.
Okay.
Mutations allegedly evolve proteins by creating new ones and by modifying existing ones so that they're different, right? But the problem is the biological evidence simply shows that when you mutate proteins, it proceeds, and evolution can't proceed by a product, can't create the thousands upon thousands of proteins in all the different cells of all the different living creatures in the world by a process that overwhelmingly causes disease, you see. So that's not rational.
And I guess I would agree with all that. I myself am a supporter, or I guess my opinion is inclined toward what's called deep homology, where I don't particularly believe in, or at least the evidence to me indicates that the genes themselves are not what changes. It's the current evidence suggests the regulatory elements are what changes, and that they're inherently less pleiotropic than, say, changing a developmental gene, and it influences that gene's function in like 5 different parts of the body. Whereas if you change one enhancer, for instance, it just changes the gene's influence in, say, arms or legs, and it's very compartmentalized. So I guess would you accept deep homology?
No, I don't believe that any of the created kinds are related to each other. So I don't believe that homology, if you're alluding that homology is evidence of common ancestry for all living things, I would say no. But the problem with homology is that you can have 2 creatures that look very similar and they not be related at all. So the evolutionist assumes automatically.
Okay, wait, foreskin.
Okay, now we're going after homology.
Well, okay, not even just that. If you want to go after like morphological not morphological homology, but morphological convergence, usually it's very superficial. Like when you have, let's just say like an ostrich and a penguin both losing flight independently. It's very clear that was just convergent evolution. And usually the times when convergence becomes way more convincing is when you're looking at lineages that are already pretty closely related. And also, that's just morphology. We have genetics still. And genetics, like convergence on the genetic level is extremely unlikely.
Right.
Especially like sequence identity and not just like protein level identity.
Yeah.
Right. And notice how I wasn't talking about homology per se. I was talking about not anatomical homology, but more so the conservation of developmental genes, mechanisms, and he kind of avoided that.
Yeah, I think Neff wants to talk about proteins when he thinks he knows what he's talking about, and when he doesn't, he wants to talk about morphology.
Yeah.
Right.
Hey guys, I can tell you that all mammalians that exist have almost identical number of genes.
Mm-hmm.
Approximately between 28 and 30,000 genes.
Yeah.
So, the poor little mouse or just some kind of very small rodent have almost the same number of genes as the human.
What's really mind-blowing about mice is I have a huge book. It's about 2 feet across of the mouse brain. And the mouse brain is so complex and so similar to human brains that it is frightening. And they're little tiny, tiny brains, right? And they have like 1,300 different parts.
They're kind of stupid, but they're doing very good one thing, running and hiding very well.
Yeah, they're kind of stupid, but the morphology, the similarity between our brains and theirs is frightening. So I think that explains creationists.
Yeah.
They have a very similar brain. Anyway, it's just an idea. It's a hypothesis.
Yeah.
Moving on?
Yeah.
There. They jump to that conclusion. And just like with Acanthostega and Tiktaalik, they're very similar looking creatures in a lot of ways, but one is an amphibian tetrapod and the other is a fish.
Wait. He said, well, it was Acanthostega and what was the other one?
Not a great difference.
It was Tiktaalik and Acanthostega?
Yeah.
And one's a fish and the other's a 4-legged tetrapod?
Yep.
Oh my God. Okay, so it's not even that big of a, he's looking at the most basal stegocephalian, right? And then the most derived stegocephalian. So they're still like way more closely related to each other than either would be to like an amphibian. Like modern amphibians are way more closely related to us, or not way more closely related to us, but we share a more recent ancestor with like modern amphibians than we would with like Acanthostega. They're both very, very basal.
I want to say something. it's very important. Otangelo, let's be scientists, okay? Don't be mean.
No,
I never been mean with you.
that's not really him.
I'm really respect anyone that is discussing with me, and I never been mean. Please.
Okay, Demeter, that's not really Otangelo.
I mean, this is kind of you destroying everything.
Well, this isn't the actual Otangelo. This is somebody who made a troll account to pretend to be Otangelo.
Yeah.
It's a sign of weakness. This is a sign of weakness. Otangelo.
Yeah, this isn't the real Otangelo. This is Otangelo Grasshole, not Grasso.
Yeah, Grasso. Let's see. What were the two that he mentioned?
It was Acanthostega and Tiktaalik.
Yeah.
And also he's ignoring the other intermediates that we have. It's not just those two anymore. It hasn't been just those two since actually it's been a long time since it was just those two. What, like 2004 or something, or was it the late 90s?
But he was saying that Tiktaalik was a fish that had limbs, I think.
Tiktaalik had a neck and it had wrist bones and a pelvic girdle.
Yeah, but he insists that it wasn't a neck and there weren't wrist bones. It didn't have fingers. He actually contradicted himself in another video.
Yeah, I mean, Tiktaalik had a huge amount of synapomorphies that align it with like modern tetrapods, right? Like it had the reduction of fin rays, and instead it had protrusions that are analogous to fingers. It had a wrist and elbows and the beginnings of a shoulder. Yeah. It had a pelvic girdle. It had a neck, which modern fish do not have necks.
Yeah, he insisted that the shoulder couldn't support weight, but it actually was getting to the point where it could support a little bit of weight because it could lift itself up, right?
Well, so the modern thought for Tiktaalik is Tiktaalik probably like lived on the bottom of like fast moving streams and stuff, and it used its appendages to hold on to the bottom. Okay. And even by the time we get to like Acanthostega didn't have the 5 digits that are fixed in all tetrapods. It had like 8 or something. And I had this study. It might have been for Ichthyostega, but Ichthyostega is fairly similar to Acanthostega, where it showed that its locomotion on land would have sucked. It wouldn't have been able to walk like normal organisms do on the ground. It would have flopped around like a seal, right? So we're seeing progressive adaptations towards land, right? It's not, one's entirely fish and the other is entirely tetrapod.
Yeah. Yeah, I'm interested in that shoulder girdle and the development along those lines. How much fossil evidence do we have of basically transitions?
A lot. I mean, so just in Stegocephalia alone, there's a huge amount. Like I said, you have Tiktaalik. Well, so the most basal would be like Eusthenopteron and other like Rhipidistions that are a little bit more derived than like modern lungfish. And then you can just trace that line all the way up through like Pandrichthyes, Tiktaalik, Ichthyostega, Acanthostega, and then you can get into like early organisms that we're pretty sure lived on land for at least some portion of their life.
Yeah. Okay, move on.
Yeah.
We're at...
So you can take creatures of different homologies and line them up and create an image of evolution in your mind if you want to. That doesn't mean you've demonstrated ancestry.
We got about like I wasn't I wasn't particularly interested in the homology per se of structures. I was more interested in, say, for instance, have you heard of the gene Distillus in insects?
Maybe, I don't recall.
So I based on my memory, the gene Distillus is also part of beetles' legs, and it turns out that the same gene was co-opted in the horns of certain beetle species, and now it helps form the little protuberances that are part of male structures. And so
I guess
when you're saying, repeat that again, in the same species? Are you saying in the same species or two different species?
Well, like, you mean what species is Distillus part of?
No, are you saying that this one gene is found in two different species?
Common design.
Yes, it's a developmental gene, so it's a part of most, if not all, bilaterian animals.
Okay, so I would say common design, just like
How do you spell that gene, Andrew?
Distillus, DISTALS, something like that.
AL and then IS?
It might have only one L, actually.
Distillus? Like that?
Yeah, Distillus.
I'm not sure. I'll try to find it. Because Wiki is really bad at it. Okay. Well, he's pulling the common design on us.
in Microsoft Windows, when you click File and Open, or File, you get a document. It's basic code, and it does some really good things. I think the creator put similar snippets of information in the genomes of many types of creatures because it's good code. It's simple, and it works, and he could use it in many places. That's the creationist view of it. But the evolutionist has a terrible burden here. They have to demonstrate that that information can arise by random mutation, and there's no scientific reason to believe that.
I'm not saying that it arose at all. I'm saying that and
right, but it would have to have if evolution were true. So because there are genetic similarities in various living things doesn't demonstrate by itself that common ancestry is true. I understand the evolutionists are going to see it that way because of their perspective, but that doesn't demonstrate it's true. The creationist perspective is common design. See, now which one of us is right? You say common ancestry, we say common design. Now we've got to get to the nuts and bolts of it and see which one of us has the upper hand. How can we show that evolution can create this? And how can we, if evolution is able to do it, and how can we creationists show that it's a product of design? See, now that's where it goes. Because it can be used, it's found in various creatures, doesn't verify that common ancestry is true. And I wouldn't say that because it's found in various creatures that creationists have been able to verify common design. But each camp can see it in that way because of their own perspective. Now, what we've got to do is find some other way of looking at it that demonstrates to us, is it a product of creation or is it a product of design? See, so both of us are going to see that as support for our own views, you see, because of our perspective. So we have to go further than that to find out which camp is correct.
So if what you're saying is correct, you're saying that creationists accept developmental genes as part of their model.
Well, developmental genes are according to creationists, are designed. You know, the organism, without them, you can't have the organism. God had to give creative developmental genes to creatures for them to even exist, right? I mean, they have to develop, and from that reproduction has to take place. There's going to have to be information that creates the organism from a single cell, from a fertilized egg, for example.
Yeah, so I guess my position would be that I think the same developmental genes are at work, and so I'm trying to find a way that we can differentiate between our two positions in terms of development. So I'm not necessarily talking about common ancestry.
Yeah, well, that's a good question. How does evolution develop a fundamentally different type of creature? And it comes back to this idea of random genetic mutation. Mutation would have to be this incredible designer, and we don't see that. We don't have the scientific evidence that tells us mutations do these fantastic things. Nobody has put forth that evidence. We don't have it, but it's necessary if evolution is true. So where is it? That's the problem for the evolutionist camp. They can't show that mutations design anything. It seems like all the biological science demonstrates that mutations are destructive, and that's the problem for them, not for us.
Right.
Because we would say genomes were designed, and they say they evolved, but if it's complex specified information, then evolution is false and creation is true.
Right, and that was kind of the point with what I call the bristle question, the question I posed to you guys, which is I'm not arguing whether or not it's a gain or a novel information, a new system, for instance. I'm asking if it's what specific part of it would constitute the net loss of information according to your position. That's my question.
Well, I'd have to know more about it to be able to state. So you're saying that a gene is found in two different species, and you're asking me how it's a loss of information. I don't know that because I don't know any more about it than this. Are you talking about the loss of bristles?
Yes, I'm
in
talking
the reproductive area?
About that gene.
Okay, so I wouldn't say that's necessarily a loss of information unless the information is turned off and never turned back on again. If that's the case, then the information has been a loss to the species, even if it exists written in the book. So if you have a book and it's read by one guy to another, but he omits a sentence, but he's conveyed everything else in the book, then the information might still be written in the book, but it's now the guy that he reads it to doesn't have some of it. So it might be physically present, but not conveyed. See, and if that happens to the population, then the population would we would say that's a loss of information because it's not expressed. It might physically still be coded in the genome, but it's not. If it's not expressed, it's a loss to the organism. It may not be physically gone, but it's a loss to the animal because it's not expressed anymore. That's what a gene is. When a gene becomes permanently recessive in a creature due to changes to allele frequencies, then that's exactly what goes on. And
wait, Fortian,
do that.
Just a quick point. That's not what recessive means. A change in allele frequency does not make something recessive.
Right.
Recessive is whether or not it's expressed.
He's just babbling at this point.
Yeah, I think he's rambling. He's using sciencey sounding words that he thinks make sense when you string them together, but they don't.
Was that distal less? DISTALLESS? Like
that's how it's spelled, I believe.
Okay, the homeobox gene. Yeah. Okay. It's driving me crazy.
All right, I think we only have a minute left here. If you want to finish,
we'll roll through it.
Yep.
Okay.
Dr. Kenneth Miller tried to tell me that doesn't happen, but he's completely wrong about that. Because if the organism doesn't express the information anymore to the species, it's a loss. Just because it's physically present doesn't mean the information isn't lost. And I think I gave an example of why that is. If it's not conveyed anymore, the animal's at a loss, has experienced a loss of the information.
Right, and I'm saying that in this particular example, the this enhancer mutation didn't affect whether or not it was expressed. It affected where it was expressed. For instance, it affected the amount of expression. And so it didn't completely switch it off. It didn't completely switch it on. It kind of did both. I'm trying to find.
Well, I mean, really, now consider you're not basing your belief on evolution or creation upon a mutation which does something like this, or you have to you do understand that if evolution is true, you have to be able to have reason to believe that mutations do far more fantastic things, like then reduce the number of bristles on the genitalia of an organism and increase the number of bristles somewhere else on it. I mean, you've got to have bristles come into being.
Right.
See, that's the thing. So really, this is kind of a side issue. It's not really getting at whether or not evolution is true by processes like this, because if evolution is true, then you have to explain the arrival of the bristle. Right, less or more bristles doesn't constitute evidence for believing that bristles come into being. See, that's the point that I would point you to. So if you're digging your head into this thing and thinking, well, this might help me understand why I'm right if I say evolution is true, then you're going about this the wrong way.
Right, and again, I'm not trying to prove whether or not evolution is true or whether or not creation is true. I'm trying to see how you guys would define the terms in terms of this example of information. Because if the information is there, if it's already pre-existing, and this mutation just changed the expression of it, then does it constitute a net loss or a net gain? That's my question. Or that's part of my question, I should say.
Yeah. Well, as I said, demonstrating that the information has been, it comes into a being is the problem for the evolutionists.
Right, and I would agree with that. I would just say that you would also have to define a loss of information.
Did you want to hear that again?
No, I think I heard it right. It just doesn't make sense.
Yeah. He stopped making sense when he started.
What that counts for. And so far I haven't been understanding it as well as I feel like if it's this foundational concept to the creation model, then there should be a stricter definition of it, and that's what I'm trying to figure out.
Yeah. Well, I think maybe your requirement that you need a stricter definition is probably an error. You're pointing to the slipperiest.
Come on, that is the. I think that's the cream on the pie, man. That's excellent. You requiring a stricter definition is an error. What else do we need to know about this motherfucker?
Example, imagine. And
go ahead.
I was just gonna say, I think Leo mentioned this in a live stream a while ago, and it's like it's extremely correct. And it's like the creationist dudes can't deal in hypotheticals because it makes them critically think about their own positions.
Right.
Yeah, they don't care for that at all. It's quite bothersome.
Just
nasty evolutionist. Well, let's roll it to the end.
And not to something that's more common, like an amino acid substitution, which results in a defunct protein. Those are the more obvious examples of how mutation destroy. Right. So to say that we don't have reason to believe mutations destroy when we have these kinds of things is just, I don't think, that's not correct thinking, I think.
Right. So well, I think we disagree on that, but unfortunately I have to head out now, but thank you for the chat. I had fun.
Okay. Thanks for coming.
Okay. We did it. Hey, we made it through a goddamn stream once in our lives. Congratulations.
Yeah, I think just to say something that I wanted to add earlier, like if well, like for one thing, he gave like 4 definitions of information in that whole part of the stream, and secondly, that if you define information as gain or loss of expression, then like I was saying, this example, if you were to say the loss of bristles in one part was a net loss, then the other part where it was gained in another region of the body must therefore be a gain. So therefore, was this a net loss or a net gain, or was it just a modification?
Right. Looking into bristle development. There's a whole lot on it. So the only thing that the only way you're going to be able to talk to Nephilim is to put his nose down in like the actual evolution of the bristles, how the bristles were basically morphed from scales or, ectoderm or whatever. And that's about all we could do. You still with us, Demeter?
What exactly you're talking about?
Oh, I was talking about so the whole thing was about bristle development in Drosophila in the fruit fly. There were two different species, and they found that a very slight mutation in a regulatory region increased the bristles in one part and decreased the bristles in another part, and then the reverse. So that was Andrew's bristle question.
Yeah.
Does that mean new information, basically? So that's kind of where we're at.
Well, now that we've actually successfully finished something, I think I'm probably going to head out. I'm feeling kind of crappy, I'm just going to probably watch a documentary or something.
Let us know how you are. What are you going to watch a documentary on?
I don't know.
There's a real good.
Usually I just watch nature documentaries. They're pretty. I don't. So there was a time, like a little over a year ago, when I watched a bunch of like. When I mostly watched like documentaries on like prehistoric life or human evolution.
Yeah.
And it sounds kind of conceited, but I feel like I've kind of gone past like being able to learn shit from watching those documentaries.
Yeah.
So I'll just look at pretty animals for now.
Yeah. Yeah. Yep.
Right.
All right.
Anyways,
Okay.
Take care and keep us posted on how you're doing.
Yeah, I don't know. I mean, I'm pretty much the ideal demographic to have it, so I don't feel that bad.
Yeah.
But
anyways, I hate you for being young.
Yeah. All right, night, dudes.
Yeah, he's actually going to live through it.
Well, I think I'm officially done with Neph.
Yeah, well, you should be. I'm surprised that he didn't talk over you a lot because he usually does. So.
Yeah.
Anyway, but we did a good job. We made it through a stream, and that was great. Demeter, so tomorrow at 2, let's crack open the genome because I have a hell of a lot of questions about introns, particularly, and conserved regions and introns, and maybe we could find a few things.
So tomorrow, I can show you in general the website, how to work with this website.
Yep.
And what is available and what is not available. And after that, you have to click a lot. You have to play with this website probably 1 month by clicking, looking, and I can show you how to do what to do. And you in your own, you can investigate everything, every single gene, everything. You can not just single gene. You can investigate any region. So
that's a UCSC genome browser.
Yeah.
Good.
You can download in any file, like a text file, the sequences. You can do whatever you want with these sequences.
Yeah.
I mean, it's a fantastic.
Yeah, I want to drill into one developmental system, get to know all of the proteins, and start comparing across species. I mean, there's some things that there's a lot of. I'm guessing that it is such a huge problem that anything anyone does will probably be something nobody's ever done before. You know what I mean?
Mm-hmm. Well, so it's kind of I mean, it depends what you want. I mean, the most important to me is your question. If you have clear question what you actually would like to do, you'll figure it out.
Yeah.
And based on what I'll show you tomorrow as a ability and possibility of this genomic browser, you can ask whatever you want, because you have the actual sequence, and not just the sequence. I mean, you have lots of features to this sequence because the tracks. So there's a different tracks to any of those. So having factors that playing a role in development, is that what you want to say?
Yeah, I'm looking for well, developmental. I guess okay. If you looked at the nuclear pore complex, and looking for homologs back, I guess all the way back to some bacteria, that would be one problem. But another one would be like limb development, like looking at the differences in the regulatory genes that affect the development of forelimbs in mammals, for one, and then to cross that into fish.
And you can do a lot. Yeah, you can do a lot. So you just need to have the list of your genes that you want to looking for.
Yeah.
And you can cross it. You can even, I'll show you another program, online program, which can do just alignment.
Okay.
Basic alignment. I mean, you can do it by yourself, probably. Just
and
you probably will be very easy to even make several rows of program to do that.
Yeah.
But alignment, and you can compare, and pretty much everything is there. Just the time is not there.
I've got about 16 projects in the
so if you have time
in the fire right now. Hi, Midnight. You're kind of late. We're just about to wrap up.
I mean, don't wrap up on my account.
Hi, Sparky.
No, we're not doing it because of you. We're doing it because we're tired.
I think Dr. Daniel was kind of like. Oh, sorry.
Well, no, I was gonna say, if you're tired, why do you continue going after these guys? I mean, no offense, but Dimitri, Dimitri's time could be better spent rather than, wasting his time, his greatness, debunking these idiots.
He loves it, don't you?
Oh, come on.
Yeah, that's why he hangs around with us guys. Us hoodlums.
Whatever floats your boat, Yoda.
So, Demeter, Tom Jump is looking for an abiogenesis researcher, but I think he's also looking for somebody that that's he said famous or something. But I think we should hook you guys up because you might be able to help him. T Jump has a channel.
Well,
and
obviously I'm not famous, but I I can discuss abiogenesis. It doesn't matter for me, am I famous or not. If someone want to discuss abiogenesis or origin of life,
Yeah.
I will be happy to discuss it.
By the way, it looks like we're not getting $2,000.
Did you receive my email with the names and publications, papers that was dedicated to origin of life research?
The what now?
The email that Immutable, he asked me to give him
A list.
names for origin of life, the scientists and papers that are kind of very important for origin of life. Immutable wanted to read those, and I composed one email, and I sent it to maybe not all of you, but to you, Mike, for sure was.
Yeah, I did see it. Yeah. Yep. Yep. That's good.
You can distribute the same email to whoever you want. Doesn't.
What is a member from Oslo?
To your judgment.
What does that mean?
Oslo, origin of life, something.
Okay. I don't know what Oslo
I'm
is.
not member of any origin of life organization.
Dr. Dan has a link for me.
Oh, cool.
Yeah. Imgur. Oh, as if I didn't have enough to do.
Okay, guys. See you tomorrow.
Okay.
Good night.
Okay. We'll see you tomorrow. I think we're about to wrap it up. What's up, Midnight? How you doing?
Oh, just enjoying my new place. It's been.
Oh, you're in your new place.
Yeah. Yeah, it's been interesting these past 4 days. Been nice, quiet, a bit lonely, but doing these streams is nice.
Yeah. Yep, we're kind of having fun. We did not like 9 hours yesterday, and I sure as hell don't want to do that now.
You did 9 hours?
I think it was 9 hours yesterday. We did like a 4-hour weather report, and then we did another 5 hours afterwards. I bopped out for an hour. I wussed out and went and watched his Dark Materials season finale, and then came back, and they were still going at it. And we had two philosophers.
Good.
We had two philosophers, and they were fighting with each other.
Yeah.
And it like my joke was, two philosophers walk into a bar. ...
If you're talking about Nikola, I don't think he'd consider himself a philosopher.
I know he doesn't call himself a philosopher, but he is, whether he calls himself that or not. I've talked to the man; he knows his shit, and so does Danny. And I really wanted those two to get along. It was like, like I was trying to like I was a mom trying to get two to set up a little mating thing, and they fucking hated each other.
I mean. Yeah, I'm pretty sure if you ask Nikola about it, he'd say, "Yeah, the other guy didn't know what the fuck he's talking about."
Yep, that's what Nikola said. Yeah.
There you go. I agree. I agree with Nikola. Like I agree with you that Nikola knows his shit because, I've heard him talk and he does.
He just has a way about him. Well, he talks a lot, so I mean he kind of dominates the conversation. Slow down.
Yeah, he doesn't stutter either, so he's able to flow. From one word to the other, and people remember mesmerized.
Yeah. Yep.
Yeah. Well, I think to wrap up our a little chat here, since Dr. Dan is the one who helped me decide on the name for the question I asked Neff, if it's okay with him, if he's still in the chat, if he want to respond to this, whether or not do you think I stumped Neff, or do you think that I need to improve the question, or what are your thoughts on that? I guess
you did a really good job of pulling him back to the subject over and over again, and I think you kind of had him on the run. That's why he was babbling incoherently about recessive genes and crap like that. And so I'm gonna download that little piece and get a transcript for it, because it was very revealing about how a creationist thinks about information. And the wonderful part was when he said that you're wrong because you're asking for a detailed definition. Because that shows how they don't think about information. They have their narrative, but they never, and they claim they read all these, like John Maddox reads all these information books. And, he's read all these papers, and he's an information specialist, he calls himself. And he doesn't know jack shit about information, either in communications, computer programming, or biology. And they're all very, very different things. The biologists are trying to wrangle this information.
Are we talking about Maddox?
What's that?
Sorry, I zoned out for a minute. Are we talking about Maddox?
Well, Maddox and who the hell else thinks they're an information specialist? Otangelo a little bit.
I mean, I'm a moron, so I don't know anything.
Yeah, and basically,
wow, you agreed with me, Speed. Wow.
Oh, I'm sorry, you are not a moron. Okay, this channel does not condone calling Midnight a moron.
No.
I'm just messing with you, having a little fun. I'm bored.
But the thing is that this whole thing with biological information and coding and all these and the extended synthesis is a developing field that's going to be happening for the next 100 years. And they're right at the beginning of it. And there's a lot of disagreement about it. And the creationists have jumped on this shit, and they have quote mined and cherry picked because they have found the fucking mother lode. Yeah, they have found the mother lode for quote mining in information theory and biology. Yeah.
Well, I think for the most part, Dr. Dan seems to agree with you, Speed. So
good. Oh, we agree on something.
Well, thank you, Dr. Dan. That means a lot.
Yeah. Normally, Dr. Dan points out, or I don't know, maybe I'm just sensitive, but he's caught me on a few wrongness things occasionally. Demeter too. I appreciate people who have been in it longer than I, pointing out where I'm wrong. So anyway, we agree on a lot. Good. Thank you.
Yeah. Sometime, Dr. Dan, we need to I still want to have our own little conversation about evolution whenever you have time, if you're still open.
Yeah. I would. Yeah, I'd love to have you on the channel again to just have a conversation about information and evolution, Dr. Dan.
Yeah, that would be awesome.
Yep. So we're doing this 2:00 weather report every day where a lot of people climb in, but we can also, like tomorrow, we're going to limit it to Demeter and maybe just a few, Andrew and Walker, but we're not going to let everybody in because we want to actually get through we want to get through this USC, what the hell is it? UCSC browser, genome browser, and learn how to use it. So, okay. So Dr. Dan wants to do that at some point.
Sure.
Okay.
Oh, yeah. That sounds great, Dr. Dan.
There's so much to talk about.
Yep. Yeah. So I guess you and I will have to set that up, Speed.
Wow, that's a compliment. Thank you. I needed that. I was feeling bad tonight. Anyway, okay.
Awesome.
Okay, well, we're going to end the stream, guys.
Sure. Yeah.
Thank you all. Thank you, Dr. Dan, for all your comments.
Yep. Thanks, Dr. Dan.
I'm going to go eat a hamburger, watch some meaningless Amazon Prime.
Yep. Yeah, I'm going to go get dinner as well.
Okay.
And I'm going back to reading interesting stuff about biology and neuroscience and all that good stuff.
Hey, cool. I got a book called The God Problem, and somebody recommended it to me. I got it for Christmas. It's about 1,000 pages, I think.
Damn.
Anybody know about this book?
No, I don't. Not yet.
Okay,
No,
I gotta keep up on that.
Speed.
It's gonna be like my bathtub reader for a while.
Speed. When you end the stream, don't leave yet.
Don't what?
When you end the stream, don't leave yet.
Okay. I'm gonna end now. Goodbye, everybody, or anybody who's still watching. Thank you all.